The urea cycle is the process of removing ammonia from the body. Ammonia is the end product of protein metabolism. It is toxic to the human body, especially the central nervous system. The body must convert the ammonia into a harmless, water-soluble compound to be removed safely from the blood. In some animals, such as birds, nitrogen is excreted as uric acid, and the liver of bony fish excrete the nitrogen as ammonia into the bloodstream, which quickly exits through the gills. Proteins are consumed in the diet and get broken down into amino acids in the stomach and small intestine. Some of the digested protein is stored in the muscles and the rest is turned into waste, which the body doesn't need. The urea cycle's job is to turn ammonia into a non-toxic component called urea, which is then eliminated. The key amino compounds entering the urea cycle are aspartate from transamination of oxaloacetate, and the free ammonia, which is produced by hydrolysis of glutamine and oxidative deamination of glutamate, as well as other ways. This ammonia must be excreted in some way. In mammals, the urea cycle ultimately converts ammonia into urea. The cycle begins with the reaction combining a bicarbonate ion with an ammonium ion to form carbonyl phosphate. Ornithine moves into the mitochondrial matrix where it combines with the carbonyl phosphate to create citrulline. Citrulline moves out of the mitochondria into the cytosol where aspartate joins to form argininosuccinate. A molecule of fumarate is lost and arginine remains. Now water combines to remove the nitrogen as urea, and ornithine is left to start the cycle again. The first step of the urea cycle begins in the mitochondrial matrix. The following steps occur in the cytosol. Ammonia is produced in the mitochondrial matrix by the enzymatic activities of glutamase and glutamate dehydrogenase. Carbonyl phosphate synthetase 1 is the enzyme that takes the ammonia, bicarbonate, and 2 molecules of ATP to produce carbonyl phosphate. This enzyme activates bicarbonate by the same method used by biotin-containing enzymes. In fact, carbonyl phosphate synthetase is homologous to the biotin family of enzymes. Carbonyl phosphate synthetase activates bicarbonate by phosphorylation with ATP to form carboxyphosphate. Ammonia then reacts with the carboxyphosphate to form a carbamate intermediate. A second molecule of ATP is used to phosphorylate the carbamate intermediate to form carbonyl phosphate. Carbonyl phosphate synthetase 1 is the first committed step of the urea cycle. As one would expect, this enzyme is allosterically regulated. This enzyme is allosterically activated by N-acetylglutamate. N-acetylglutamate is a precursor for ornithine biosynthesis and is an essential activator for the urea cycle. N-acetylglutamate also serves to coordinate related pathways. The carbonyl phosphate formed by this reaction enters into the urea cycle as a substrate for ornithine transcarbamylase, which catalyzes the reaction. There is a citrulline transporter that transports citrulline from the matrix of the mitochondria to the cytosol. The next step of the urea cycle is catalyzed by argininosuccinate synthetase. This enzyme uses ATP to activate citrulline by forming a citrulline AMP intermediate. In the second half of the argininosuccinate synthase reaction, the alpha amino group of aspartate attacks the amino group, releasing AMP and producing argininosuccinate. In the mitochondria, oxaloacetate is transaminated by glutamate by aspartate aminotransferase to produce the aspartate, which is then transported into the cytosol where it is required for the argininosuccinate synthase reaction. These reactions together are called the aspartate argininosuccinate shunt. The third step of the urea cycle is catalyzed by argininosuccinate lyase, which catalyzes the reaction. The products of this reaction are arginine and fumarate. The fumarate product is an important link between the urea cycle and the citric acid cycle because fumarate is an intermediate in the citric acid cycle. The last reaction of the urea cycle is catalyzed by arginase, which catalyzes the hydrolysis of the guanidino group to produce urea and ornithine. Ornithine is then transported back to the matrix of the mitochondria, and the cycle is complete. There are two nitrogens in urea. One came from the ammonia produced in the matrix of the mitochondria, captured in the form of carbamoyl phosphate. The second nitrogen was the alpha amino group of the aspartate substrate in the argininosuccinate synthetase reaction. The sole carbon of urea was originally the carbon of bicarbonate. The urea path through the body. The urea cycle occurs in the mitochondria and cytosol of liver cells. About 75% of the urea travels through the bloodstream to the kidneys. The kidneys filter the urea and excrete it in the urine. The other 25% of urea is sent to the colon, where it is converted back into ammonia and carbon dioxide. Some of the ammonia is excreted in the feces and some is sent back to the blood. About 85% of ammonia sent to the blood will go back through the urea cycle. The other 15% will go into the systemic circulation. Hyperammonemia is the presence of an excess of ammonia in the blood. A defect in the urea cycle can cause accumulation of ammonia and other products. This can result in encephalopathy, coma, and death if not recognized and treated rapidly. Diagnosis of urea cycle disorders can be confirmed by testing for excess ammonia in the blood in suspected patients with neurological symptoms in the intensive care setting. Treatments available are based on the removal of ammonia by dialysis or hemofiltration, reduction of the catabolic state, abolishment of nitrogen administration, and the use of pharmacological nitrogen scavenging agents. Enzyme deficiencies in the urea cycle. Argininosuccinase deficiency is when levels of arginine drop, decreasing the amount of urea formed. For example, a genetic defect caused by a deficiency of argininosuccinate lyase can be partially overcome by providing a surplus of arginine and a restricted protein diet. The surplus arginine is converted into urea and ornithine by arginase. The ornithine produced reacts with carbamoyl phosphate to form citrulline. Citrulline is then condensed with aspartate to form argininosuccinate. The concentration of argininosuccinate builds up and is excreted. In this way, argininosuccinate substitutes for urea in carrying nitrogen out of the body. Other deficiencies include a deficiency of CPS1 or ornithine transcarbamylase. The ability to synthesize citrulline is impaired. Under these conditions, excess ammonia accumulates in the liver and kidneys as glycine and glutamine. The challenge is to get rid of these two amino acids. This is accomplished by supplementing a protein-restricted diet with incredibly large amounts of benzoate and phenylacetate. Benzoate is converted into benzoyl-CoA, which then reacts with glycine to form hippurate. Likewise, phenylacetate is converted into phenylacetate-CoA, which then reacts with glutamine to form phenylacetylglutamine. These two compounds can then be excreted to partially overcome the genetic defects. Dealing with excess ammonia in the body, glutamate can be catalyzed by glutamate dehydrogenase to form alpha-ketoglutarate as well as the free ammonium ion. Alpha-ketoglutarate with the free ammonium ion can then form glutamate. The problem is you must get this alpha-ketoglutarate from the TCA cycle, therefore ATP is lost. With glutamine, the glutaminase will form glutamic acid and a free ammonium acid ion. Glutamic acid with the free ammonium ion forms glutamine. Both processes are very energetically intensive. Regulation of the urea cycle, diet, and CPS1. Dietary protein and CPS1 is the first committed step or the rate-limiting step of the urea cycle. This enzyme is allosterically regulated by N-acetylglutamate. After ingestion of a protein-rich meal, the intrahepatic concentration of N-acetylglutamate increases. This drives the urea cycle, ultimately increasing the rate of the urea synthesis.